All right, in the second video of our video series where we break down the assessment notifications, we have Oran Anglican's College and the first biology task for year 12. And it's a modeling task. So I've noticed that most schools this year for year 12 bio, they've been given the polypeptide synthesis modeling task. And let's kind of break it down. So in the assessment task, it says that students are required to construct a physical model in the form of a storybook, demonstrate the process of polypeptide synthesis of the following peptide chain his, glin, and tyre. Compile and provide a bibliography, answer written questions related to the use of scientific models, DNA replication, polypeptide synthesis, transcription, translation, polypeptide formation, and mutation by the due date. Why is it important? Okay. All right. So just very quickly, before I go into the notifications, like exact marking criteria, you'll have to talk about polypeptide synthesis, and you'll have to mention translation, transcription, and for each of these different parts, you have to clearly show that you understand it. So for DNA, you have to write something. You want to create a model which contains the sugar and the phosphate. You have to mention phosphate, you have to mention sugar, provide a backbone. And you can either use beads for this or buttons. Beads/buttons. Just note that your model is going to be stuck onto a piece of paper. And for your bibliography, you'll find that most of your information is in textbooks or it's in things that you already have come across. Maybe if you go to tutoring, then most of the time they'll give you a lot of the notes that you need to do tasks like this. You can search online. There's plenty of YouTube videos. There's plenty of like scientific papers that kind of go over these things, or like even magazine articles online, and you can use that for your bibliography. It's very difficult to reference your tutoring notes because technically they're not published, so it's difficult to reference that. Okay. Something else you have to know is that these are your 3 amino acids. So you want to take the amino acids and translate them backwards, and just move backwards to try and translate them back to the DNA strand that it should be. So let me just write this out. So if I have his, then GLN, then TYR, and that's my amino acid chain. Let me go back. Let me go to the table of amino acids and mRNA molecules and figure out exactly what I should write down. So let me get rid of this bit. All right, it's a bit higher. His, GLN, TYR. And that's your amino acids. Okay. Now, if you're not really familiar with this is how you translate or kind of figure out what a amino acid, so basically which amino acid is going to form given an mRNA code. Now, since you have to take this amino acid chain or this polypeptide chain and then try and model from when it's DNA all the way to this point where it turns into these amino acids, you're going to have to use this table. Now, all amino acids begin with a start codon, and the main start codon is MET. So the amino acid MET is the first amino acid that's made in pretty much every single polypeptide, and that started off with AUG. So in my mRNA, it's going to be AUG. That's just for MET. Cool. And then for HIS, if I go and find HIS, it's over here, I can see that there is a couple of different ways for me to model this. It's either CAU or CAC. So let's use CAU. Then GLN, that's over here, CAA. And then TYR, UAU. Now, technically, this should also contain a stop codon, because these are my main amino acids. This is just, met. Every single polypeptide has this, but like, I need a stop codon. I need something to tell my ribosome to stop coding for these amino acids. So I need to pick a stop codon. So maybe I'm going to use UAA. Okay, so that's my mRNA. Now for my DNA, it's going to be the exact same thing. Specifically, like, the U's are going to be converted into T's. So imagine this as your DNA and specifically your coding strand. So AUG is going to become ATG. CAU is going to become CAT. CAA is going to become CAA, because it doesn't really change. UAU is going to become TAU. UAA is going to become TAA. So think about that as your coding strand. So that's like the overview, the very beginning of this assessment task. And now you have to think about how you're going to model this. What are you going to use to represent the nitrogenous bases? What are you going to use to represent each of the different parts? So let me just quickly skim through the notification guidelines, and then we'll use this to kind of build out a model and kind of predict, or kind of just make a model on here so you can get an idea of what materials to use and how your model should kind of look like. Okay, 9 to 10 aspects of protein synthesis: DNA, mRNA, tRNA, ribosome, amino acids, nucleus, nuclear envelope, DNA double helix. So this school's been nice enough to give you basically all the answers. Like, there's not that much guesswork involved in making this model. DNA double helix, nucleotide composition, names of 4 bases, pairing, bonding between them. Okay. 5' and 3'. DNA molecules, coding strand, non-coding strand. Sequence is correct. Errors. Okay. Where translation happens, where transcription happens, enzymes located. Okay. All right. Seems okay. Let's zoom out and let's build out this model. Now, where I've seen this model being done very well is when people use beads or buttons to represent phosphate and sugar, or when people use those sticky notes. Those, if anyone uses these to do English, you might see them as like rectangular shaped sticky notes that you can stick onto a page. You can write down notes. It's almost like a mini bookmark. I'm pretty sure they're just called sticky notes. I'm not sure there's any particular name for them, but they're just the sticky notes that you use for that. So it's relatively small rectangular sticky notes. Okay, so say you wanted to build out a model for this. The way that you want to build this out is you want to have a coding strand, which is, let's say, ATG, CAT, CAA. So also maybe we can have it like some sort of shape for P, for phosphate. It's connected to a sugar, and that sugar might be a button, might be a bead, just like the phosphate, but a different color, so we can show that they're different. And then you'll have nitrogenous bases. Okay, let's assume that this part here is the coding strand. And this one here is the non-coding strand. All right, so in the coding strand, I'm basically going to write in all of these from top to bottom. So A, T, G, C, A, T, and just continue on with this. So C, A, T, A, U, and T, A, and one more A. Imagine each of these is just a sticky note. And you can do different colors for different letters. So for adenine, maybe you want to make it like a pink sticky note. For thymine, you can make it like a different color. As long as they're different, it should be okay. Make sure that they're complementary to the base pairs. Cool. And for the rest of this, you're really just going to draw out more or less the same thing over and over again. And maybe you can use pipeliness to show the connections, or you can kind of draw them in onto your page. Whatever works. Whatever you feel is the best. But here's how I've seen people get pretty good marks. You keep it relatively simple, and even if you do make a small mistake on there, they usually ask you a question regarding limitations of your model, and you can talk about that mistake there. But due to the very strict guidelines for this, I'm going to have to make the model as close to perfect as possible. Coding strand, non-coding strand. And just for just for us to save time, just imagine that all of these are connected with S's to P's, just like this. And at the end, let's say it's a overhanging S. So if there's a sugar at the very end and not a phosphate, then this is a 3' end. 3' means that a sugar is ending there. 5' means that a phosphate's ending there. So for this one, imagine this is T, T, A, and I'm just going to do this complementary. I think I accidentally wrote U here. It should be T instead. Oh, actually it's supposed to be U, A. TAA. Okay, yeah. Oh no, it should be TAT. TAT. Yeah, so TAT. For what I was transcribing this one. Okay, TAT, TAT. And this side is going to have a sugar that's ending in it. That's going to be 3'. And it's going to have more or less the same structure. So all of these are going to have the same structure as before. With the phosphate in between. And just imagine that it continues on, and then when you get to the end, it's a phosphate that ends there. So it's a 5' end. So that's how they want you to model the, at least just what the DNA looks like before it becomes translated and transcribed. And you want to have a key where you describe that, a certain, this thing is adenine, this thing is thymine, guanine, and cytosine. And then you want to show transcription and translation. So we would take this. So we want to make a mRNA copy of the DNA strand. So we want to make an mRNA copy of this strand right here. So you probably take it off to the side. Let me just zoom out a bit more. And take it off to the side. And just to keep it a bit more simple, because we've already kind of shown the marker that we understand that there's a sugar and phosphate backbone. We can kind of simplify it by putting a straight pipe cleaner down and then just drawing out each of these parts. Okay. So since I want to make a copy of the coding strand, I have to make a complementary strand to the non-coding strand. So I'm going to take the non-coding strand here instead. So we TAC, GTA, TAC, GTA, GTT, ATA, and ATT. And once again, I'd have my sticky notes representing these letters. And that's my non-coding strands. And I would show my mRNA forming. So I'd use a different color for my mRNA. Let's just use pink. So recall that mRNA, the mRNA molecule, it's going to be basically, well, in order to make a mRNA copy of the coding strand, it has to make a complementary strand to the non-coding strand. So T is going to be A is going to be T, but because it's RNA, it's going to be U instead. So G, C, A, U, C, A, U, A, U, A, and A. And notice how it's pretty much the same as what we have over here. Cool, so this is your mRNA, your messenger RNA. And this is your DNA. Do you want to be a little bit more like descriptive and add a bit more to your model? You can also show that the sugar in RNA is different. It's a ribonucleic. It's a ribose sugar instead of a deoxyribose sugar. And you can show that by having either a different color button, different color bead, or a different shape to represent the sugar there with the phosphate. But let's assume that isn't necessary for this for now. Okay, so you have all this taking place, but this is taking place in the nucleus. So you want to show that this is in the nucleus. So maybe you can have an outline on your page that shows that it's in the nucleus. And the nucleus, at least in my opinion, kind of reminds you of something that's blue. So I'll just show that this is all the nucleus. Cool. And then I have to show that it's somehow leaving the nucleus. So this is in nucleus. This is my transcription. And when it leaves the nucleus, I have to show that it's leaving through a hole. And that hole is called a nuclear pore. And then the mRNA, it joins the ribosome. It goes into the ribosome. And just before I forget, you also have to show that there's an enzyme working here. And the main enzyme in polypeptide synthesis, or in transcription, is RNA polymerase. So imagine RNA polymerase is moving this way and it's adding in the bases. And RNA polymerase unzips and it adds in the complementary RNA bases to the non-coding strand. Okay. Let's make the ribosome here. This is my mRNA molecule, which has left the nucleus and gone into the cytoplasm and specifically into a ribosome. So this is your large subunit of your ribosome. Subunit is just a fancy word for part. This is your small subunit. And there's a reason why I've arranged everything in threes, and that's because in transcription and translation, these lots of three code for a single amino acid, as you can see here. AUG, CAU, CAA, UAU, and UAA. And there has to be a structure which temporarily binds to the mRNA and then deposits the amino acids, and that structure is known as a tRNA. So tRNA molecules, they look like this. So this is a tRNA, or transfer RNA. They exist in the cytoplasm, they're carrying around an amino acid, and when they're ready and they're called upon by the ribosome, they go down and they temporarily bind to the ribosome, and to the mRNA strand in the ribosome. And then they deposit the amino acid. So notice how you have a set of three, which is known as a codon. So this is a codon, a set of three nucleotides. And the opposite of a codon is an anticodon. So this is an anticodon. So G, U, A. And it's complementary to whatever I have there. And personally, for my diagrams, I like to represent different amino acids as different shapes. You might choose different materials, different shaped materials, different colored buttons, different colored beads, whatever you like to show that they're different amino acids. I actually draw these amino acids slightly bigger. And let's say this is like a rectangle. Cool. So you have your amino acids, but they're not connected yet. In order to connect them, we need to show that they're joined up. And this joining process, let's use a slightly different color. White look good. There we go. They're joined together by peptide bonds. And you can either use string or you can use some pipe cleaners to show that they're joined together like that. So when you have multiple peptide bonds, you have a polypeptide. So that's why this is called a polypeptide. You have multiple peptide bonds. And if you want to, you can also add in the names of each of these amino acids. And notice how I haven't actually, made an amino acid for like UAA. That's because UAA is a stop codon. It just tells the ribosome to stop coding for amino acids. So I might just add in the names. METs, GLN, TYR. Oops, missed his. His, GLN, TYR. Cool. And I kind of squished everything into the side here, but you might also want to have an outline to show that this is taking place in the cytoplasm. So you kind of outline that this is taking place in the cytoplasm. Okay, this should be most of the things you need to do to get full marks on this test. I recommend just going over everything briefly again. I highly recommend using either beads or buttons to show that there's a sugar phosphate backbone. Having one side, having a phosphate and the opposite end of that strand, having a sugar at the end of it. Obviously, you're going to have to put in the rest of the phosphates. I just wanted to kind of skip a few things just to save us some time. And use those sticky notes with different colors as your parts here. You can also add the hydrogen bonds to know that between A and T, there are 2 hydrogen bonds. So everywhere there's an A and T, you can somehow show that there's like 2 lines connected them. And between G and C, there's 3 lines connecting them. So there's 3 hydrogen bonds there. So the real main part of your model is actually just this DNA structure, then the structure of the non-coding strand next to the mRNA, and how the mRNA is creating this complementary strand, RNA polymerase, which is being shown to be creating this strand, then the mRNA leaving through the nuclear pore in the nucleus, going into the cytoplasm, and then going specifically into a ribosome. So let me just very clearly label this as a ribosome. And then the tRNA molecules coming down, depositing the amino acids, forming peptide bonds, and creating this polypeptide. So this is how I would approach it. So pretty much all polypeptides start with MET, so you'll have to put that down to draw this out. Okay, hope that helps. If you guys want me to do your assessment notifications, just send it through to the email in the description box below.